Um, Hopefully that works. Okay. Hey, how's it going? I should warn you, there is like a 2 minute, not 2 minute, 2 second delay between what I say and what you hear, but you know. Alrighty. So, how's it going? How we feeling? Hey, Valentina, how's it going? It is early in the morning, but let's get to it. So, what are you guys thinking? What are we trying to look at? What are we the most slash least comfortable with? Hey, Sophie. Sophie is one of my biggest fans, I swear to God. All the comments, I love it. Thank you, thank you. You guys have no idea how long it took to set this thing up. I thought it was just as easy as like, streaming it, but turns out no, not really. I had a whole bunch of stuff to do. I had to do a bunch of test runs last night. Yes, you do comment each time. Can you see everything fine? Can you hear me okay? Okay. All right. Amazing. Amazing. Um, Again, there is like a 2 second delay between what I say and what you hear, so, keep that in mind. Um, so it's the midterm's going to be just fats and proteins this time around, right? All fats and pretty much all proteins, no vitamins. What concepts do you guys think you want me to go over? Or do you have any questions about specifically? Because, again, this is just going to be kind of really casual, right? Lipid absorption and transport in the blood. hmm hmm So I got two lipids here. Let me see which one I want to pull up. So I have, on my end, I have the slides and I have my own kind of notes that hopefully we can kind of see things. Let me see something. Do I have other ones there? I do have this one here. Okay. Um, okay. So lipid absorption specifically. So we're okay about how they get digested, right? Happens at the level of the oral cavity, at the level of the stomach, and mostly at the level of the duodenum. Okay, good. So the absorption of them is essentially once we have for example, our cholesterol molecule, right? We usually ingest cholesterol in the form of a cholesterol ester, and we ingest phospholipids, we ingest triacylglycerols, and all of this stuff, once it has been fully kind of cleaved um, at the level of the duodenum, it will break down to its kind of simplest constituents. So things like just cholesterol or just free fatty acids or um, you know, diacylglycerols and monoacylglycerols, all of this kind of stuff will eventually aggregate to this negatively charged aggregate called a micelle. All a micelle is literally just a cluster of all of those digestion products, okay? And it is lined by a whole bunch of phospholipids on the outside. All right? Does that make sense? Again, I hopefully it does. Let me see if I can change this to live chat. There we go. Okay. Any questions on that so far? No? Okay. Okay. So then once we have these micelles, right? These micelles, they're very, very small and they can fit between this regions of the enterocyte's brush border. And actually, as they approach the brush border membrane, they kind of disassociate. Okay? So what I've gone ahead and done is I've kind of zoomed in on this interaction here, and we can see that as the micelle in red, which contains all of this kind of stuff in it, the cholesterols, the monoacylglycerol, the broken down phospholipids and what have you. All of this stuff is in the micelle, and once the micelle approaches close enough to the enterocyte brush border, it starts to kind of disassociate. Okay, and all of the contents that were inside of it are going to come in here in these regions of the brush border membrane, and then they will get absorbed by their specific transporters. Any questions? Can you hear me fine? Yes? No? Okay, good. Now, there's two ways that your fats can get absorbed once they are here. They can go through a protein dependent model and a protein independent model. Okay, and I do believe that is something on your slides as well. You are taught that, right? Correct me if you're not. I just want to make sure you have this in here. Yes. So it's a protein dependent model and a protein independent model. The protein independent model is just simple, good old diffusion. So you have free fatty acid, and the free fatty acid can go through the lipid bilayer of this enterocyte without any help, just from a high concentration to a low concentration. Why would it be in a high concentration here? Because you have just ate, and so all of the free fatty acids or any other kind of lipid digestion products would be in a higher concentration outside than inside of the cell, and so they would, freely diffuse into the cell down their concentration gradient. No help of a protein transporter needed. But um, protein transporters can also play a role. So, for example, this guy right here, CD36, okay, is a free fatty acid transporter. Okay. So it may take things like long chain fatty acids, it may take things like monoacylglycerols, okay, and they will transport them in here. Okay. Um, that's the protein dependent model. Again, there is no kind of enzymatic activity going on, right? It's just a channel that allows for the diffusion of um, these digestion products into the cell, into the enterocyte. Whereas the protein independent model, there's no protein channel, they just kind of go through the membrane. Um, Cholesterols, like this right here, will use this specific transporter. Hey, Mario, man. They'll use something like this, like NPC1L1. Okay, it's a cholesterol transport. All right, so it will only take cholesterol. And again, we see that the cholesterol is coming in here through that guy. It's just another kind of channel. All right, does that make sense so far about all of that? Any questions? Anything I'm not kind of hitting the point on that you may want to ask a question on specifically? Mm-hmm. Mm-hmm. Okay. So now you got to remember that is long chain fatty acids. Okay. There is an exception that does occur, and I do wonder if I've shown it here. Maybe I have. Yes, I have. So things like short chain fatty acids, they do actually bypass the lacteal, right? So to reiterate, anything that we have digested, right, especially coming out of these micelles, will get absorbed into the enterocytes by either kind of the protein dependent or independent model, right? And they will, inside of that enterocyte, be re-esterified, all right? Does everybody know what I mean when I say re-esterified? Yes? No? It's okay if you don't. You can just ask me and I can tell you. Okay. All right. So re-esterified is you have a fatty acid, okay, and you want to join the fatty acid back to your glycerol molecule, okay? So if you remember, let's go back. Here's my molecule of glycerol, and here is a fatty acid, right? Everybody see how this is a fatty acid? It has this methyl end, it has the carboxyl end, right? And we know that we can number this from either the methyl end, which is kind of going to be the omega system, or we can number from the carboxyl end, which will be our delta system. But either way, this is the fatty acid. Now, when I attach this fatty acid to my glycerol molecule, I get this process of esterification. Okay, so throughout the process of digestion, I had a molecule of triacylglycerol, right? Well, what did I do to this molecule of triacylglycerol? I started cutting the free fatty acids bound to it. Okay, so I started kind of cutting this bond and this bond and this bond and releasing free fatty acids, not bound to the glycerol. But inside of the enterocyte, after we have kind of absorbed everything from the duodenum lumen, if you will, what the enterocyte is going to do is it's going to re-esterify, okay? So it will join back all of these cleaved free fatty acids back onto this glycerol molecule and now make a triacylglycerol again, okay? And it will do the same thing for other things, such as cholesterol, right? Remember, we had our cholesterol ester and we cleaved that into just cholesterol. Well, we're going to make a cholesterol ester again, right? So again, everything is essentially going to go back to what it was before we um, started to, you know, um, hydrolyze them. And then we're going to package them into our good old chylomicron. Okay? And then this chylomicron will leave into the lacteal. If you guys remember, lacteal is a specialized lymphatic vessel. And these lymphatic vessels is where the chylomicron or our fats will essentially drain into before making their way into the blood. Okay, so first they leave the enterocyte, go into the lacteal, which is a lymphatic vessel, and then from the lacteal they eventually end up in the bloodstream. All right, amazing. Um, and notice they actually bypass the liver, right? They bypass the liver. Whereas if you think back to things like amino acids, things like carbohydrates, they directly go to the portal vein, meaning they directly, the first organ they will see is going to be the liver. But the fats, that's not true for them because they kind of skip the portal vein and drain into the lacteal. They see the liver later on. Okay, so they're bypassing the liver. And that actually allows for this kind of chylomicron to kind of make rounds in the bloodstream and distribute fat towards tissues after a meal for either utilization or storage. But like I was saying, it's really these kinds of fatty molecules that will get packaged into a chylomicron. Whereas a short chain fatty acid, because it is really tiny, really small, it is very soluble. And so it doesn't really need to be packaged into these chylomicrons, and instead it can just go out into the bloodstream on its own. And in the blood, it can be bound to this molecule of albumin. Okay? Here's your short chain fatty acid bound to the albumin and being carried. Okay? Does pancreatic lipase break down the triglycerides inside the fat droplets as well as break down the bile salts around the micelle? So, good question. The pancreatic lipase is only going to break down the triacylglycerides inside the fat droplet. It won't touch the bile salts. Does that help? And it has to do with, the specificity of the pancreatic lipase enzyme. Um, it likes to cleave triacylglycerides at a very specific location, and it doesn't have any kind of specificity toward the bile salt. Okay, great. So, any questions so far regarding this kind of lipid transport and absorption so far? All right, all right. So I know, let me kind of scroll back up here. Um, there it is. Okay. So I know, Insia, would you like me to also kind of go through the, um, you know, chylomicrons, LDL, HDL, all this stuff, or was this specific portion just that um, one that you were looking for? Okay, okay, okay. Um, from the time the tricycle and bacon is ingested to the time the okay, so this is about it. That's a great question, and um, Mario asked me this question before. Um, there's two ways you can look at it, and this question was actually the question that he brought up in lecture. From how many times does the triacylglycerol in bacon get um, hydrolyzed and esterified until it makes it to the muscle? That question was also on our exam back when I took it, and there's a couple of ways you can look at it, but only the way he likes it is the right way. So technically speaking, there are multiple rounds of hydrolysis and multiple rounds of esterification that occur. But what he does is he has grouped all of the hydrolysis of the triacylglycerols that occur here and here to one. Okay, so he has grouped them as one. You might be asking me, okay, well, why would he do that? Because the triacylglycerides at the level of the oral cavity in the stomach, only like 30% of them get cleaved, and 70% of them get cleaved at the level of the duodenum. Okay, so majority of the hydrolysis occurs at the level of the duodenum, but he just kind of smushes those two together and says one. So triacylglyceride will get hydrolyzed once if we combine both of these reactions that occur. Everybody with me there? Okay, so where is the next hydrolysis that's occurring? Well, the next hydrolysis occurs over here at the level of the tissue, right? And specifically, since the question asks for skeletal muscle, we will be looking at this dude over here. We know that when the um, fat is digested and absorbed, it will be put into the chylomicrons, right? So we saw that the triacylglyceride will break down to monoacylglycerides and long chain fatty acids, and then they will be put into micelles. Then the micelles will be absorbed. Um, well, the contents of the micelle will be absorbed. And then we will kind of re-esterify everything, okay, back together at the level of the enterocyte. So that is one esterification here and one hydrolysis at the entire kind of levels of this. Now, once the fat is inside the chylomicron, it will move into the bloodstream, right? And it will find its way at the capillary beds of your skeletal muscle. And over there, this enzyme that sits inside the capillary called lipoprotein lipase cleaves the triacylglycerides that is inside this actual chylomicron and generates free fatty acids for the use of skeletal muscle. So that is a second hydrolysis that's occurring. All right. So to answer your question of um, how many cycles of hydrolysis and esterification does a triacylglycerol bacon go through until it makes it into the skeletal muscle and is oxidized? The answer is two hydrolysis and one um, esterification. The esterification occurs at the level of the enterocyte, right, inside the enterocyte, and the hydrolysis occurs one in the digestive tract and one at the capillaries. Okay. Now you might be asking me, well, hold on a minute. Doesn't the fatty acid that comes into the cell, from this lipoprotein lipase, doesn't that get esterified as well? And the answer is not really, because the question is asking for oxidized in the skeletal muscle. And even if it didn't say that, skeletal muscle doesn't really store that much fatty acids. It is really the job of your adipocytes to do that. And so, because of that, we assume that the free fatty acid will be getting oxidized in the skeletal muscle. Okay? Does that make sense? Does that help answer the question? Any other confusions, any other clarifications that I can maybe make for you guys there? I know it can be a bit convoluted. Okay. Let's do this. Okay. Amazing. Amazing. So, um, since we wanted to continue from and again, feel free to kind of, drop any questions you guys have in the chat, right? I can go through them. Um, we're gonna do like an hour and a half, so maybe like 12:30, we'll call it. Um, or earlier if none of you guys have any questions. But either way, let's continue through this pathway. So once we have our chylomicron inside of the bloodstream, right, it has a couple of proteins on it, and these proteins, they are called apolipoproteins. Okay, apolipoproteins, or sometimes for short form, they're called apoproteins. Okay, now these apolipoproteins have multiple functions, okay? They provide water solubility to these chylomicrons. That's one. Two is that they act as ligands for activating specific receptors that the chylomicron may come into contact with, or um, they act as activating enzymes in the bloodstream, okay? So these apoproteins are very important and they have diverse roles depending on what kind we have. Okay, so um, yeah, okay, so we have multiple types of them. We have ApoBs, we have ApoA, ApoC, ApoE, and all of these have many different kind of isoforms. All right, meaning they come in different um, flavors, if you will. All right, so when this chylomicron first starts out, right, it circulates in the bloodstream with ApoA1 and ApoB48 on it. Okay, and ApoB48 is how we can kind of recognize that this is a chylomicron on it. This apoprotein is the characteristic apoprotein of this chylomicron. Okay, and it circulates until, it interacts with an HDL molecule in the circulation. And through a specific kind of interaction, this HDL molecule will pass on ApoC2 and ApoE to it. Okay? So we'll receive ApoC2 and ApoE from it. All right. Now, once it has this ApoC2, and this is the key part, it must have the ApoC2. Once it has the ApoC2, guess what? It will interact with this enzyme called lipoprotein lipase, right? The name should be making sense to you guys. Chylomicron is a kind of a lipoprotein, right? A lipoprotein lipase is going to cleave the lipids inside the lipoprotein. Okay? What kind of lipids make up the chylomicron? Mainly triacylglycerols and cholesterol. But of course, there are other things, all right? But mainly it's the triacylglycerols, and the lipoprotein lipase is acting on those triacylglycerols, okay? Um, so once it has this ApoE and ApoC2, the ApoC2 can actually cause a docking of this chylomicron next to this LPL, and it will cause the activation of this enzyme, lipoprotein lipase, and this lipoprotein lipase will start kind of cleaving all of the triacylglycerols in this chylomicron, and that will generate free fatty acids and maybe some monoacylglycerols, but essentially these free fatty acids will go inside of the tissue's capillary bed. Okay, so for example, if the tissue that we were doing this at was the skeletal muscle, then this lipoprotein lipase would have been on the capillaries of the skeletal muscle. Okay? If it was at the heart, then the lipoprotein lipase would have been at the capillaries of the of the heart. If it's at the adipocytes, then again, you get the idea. And so this free fatty acid, depending on the kind of tissue it is, if it's an oxidative tissue like the muscle or the heart, it will go in there and become oxidized. If it's a storage tissue such as your adipocyte, it will go in there and become stored as fat, as triacylglycerol, in fact. Okay? And why will it get stored there? Because again, adipocytes are your main kind of storage cells for fat. And if the question were to ask you, um, if we go back to that kind of same question as before about like the bacon and the triacylglycerol, if we changed it from skeletal muscle into adipocyte, then the answer might change and be something like 2 and 2, right? 2 rounds of hydrolysis and 2 rounds of esterification, because adipocytes are storage cells for fat. And so when they take this free fatty acid inside of them, they will esterify it back into triacylglycerides, okay? Especially during this kind of feeding period. Does that logic make sense to everybody? Feel free to, let me know in the chat, right? Like if it doesn't make sense, if you want me to go over something again, right? Or repeat something, completely kind of, be comfortable. Okay, amazing. Amazing. So, after multiple rounds of this kind of, cleavage and whatnot, right? A lot of the chylomicrons inside of the bloodstream will start turning into what we call chylomicron remnants. And the reason they're called chylomicron remnants is because they have shrunk in size, because initially they had a whole bunch of triacylglycerols in them. And over time, as we kept going through this kind of cleavage cycle, right, these chylomicrons are getting smaller and smaller and smaller because the constituents inside of them are getting lesser and lesser and lesser. And so we have this chylomicron remnant now, and this chylomicron remnant in circulation will again come in contact with this HDL molecule, and it will give an ApoA1 and ApoC2 back to this guy. Notice though how it keeps the ApoE, and there's a reason for this. There's a reason that it keeps the ApoE. It gives the ApoC2 back because, of course, the ApoC2 was for interacting with the lipoprotein lipase, but the chylomicron remnant has already done that, so it does not need the ApoC2, so it gives this ApoC2 back. And it continues on to the circulation after. And it continues on and on and on until it finds its way to the liver. Okay? And a liver cell is called a hepatocyte. So just as a muscle cell is called a myocyte, a liver cell is called a hepatocyte. And these chylomicrons, they will find their way towards the liver, and you don't have to know this specific kind of name of the receptor, but by way of receptor-mediated endocytosis, this chylomicron remnant will be uptaken inside of the liver. Okay, so yeah, on my kind of thing over here, it doesn't mention the name of the receptor, so don't worry about the name of the receptor that I have here. All right, but some receptor R will uptake this chylomicron remnant into the liver cell, our hepatocyte, and there is an enzyme very much so close to this receptor. It is called hepatic lipase. It should make sense that this is called hepatic lipase because hepato referring to the liver, right? So the hepatic lipase is going to cleave all of the kind of constituents that were inside or remaining inside this chylomicron remnant, right? So the little bit of triacylglyceride that was left, maybe any kind of cholesterol esters or whatever, this hepatic lipase will kind of cleave those things and result in generation of free fatty acids, glycerol, and um, that's it. Okay. So is that making sense so far about the chylomicron cycle of things? Any questions? Oh, right. So the ApoE, I did mention that there's a reason this ApoE is kept, is because the ApoE is the one that has to interact with this hepatocyte receptor in order to initiate the endocytosis of the chylomicron remnant. Okay, so the taking in of the chylomicron remnant, it has to kind of be initiated by this ApoE interacting with our receptor. Okay, so that is why it needs that ApoE there. All right. Um, any questions? Does that answer your question, Insia? Okay. Um, now let's see. I want to see if there is anything that I could add on to this. Okay, so when the chylomicron remnant comes into the liver, what happens to the liver? Well, when it gets endocytosed into the liver cell, the liver cell breaks down anything that was essentially making up this chylomicron. Okay, and that's just kind of going back to regular basic cell biology. Anything that you bring into the cell has to be broken down, all right, to its tiny constituents. So all of these apoproteins that were on the surface of this, what are these apoproteins made up of? Amino acids, right? And so once this chylomicron comes into the cell, we will break it down to all of its constituents. So we will get fatty acids out of it. We will get amino acids out of it. We will get glycerol out of it, right? So we showed these, but I didn't put amino acids because in this specific video I was focusing on um, fats. But you do realize you do get amino acids, right? So what now? What happens? Is that it? Is this um, that's the only way I ever get fat to my tissues? No, right? The liver participates in something called endogenous lipid transport. So if I could go back here into this image, here is my chylomicron. The chylomicron is what we call as being part of the exogenous lipid transport system. What does that mean? It means it takes outside lipids, right, dietary lipids, and it distributes them to the body. Whereas the liver synthesizes the molecule of VLDL and it is involved in endogenous lipid transport. Okay, so imagine this. How is it that the rest of your body is receiving fat or fatty acids, right, for fuel and energy metabolism whenever you're not eating? Well, there's many ways, right? The adipocytes, for example, can kind of cleave their fatty acids and put some out into the bloodstream, but that's not the major way that you get this kind of stuff. During resting periods, you have this endogenous lipid transport running continuously to make sure adequate distribution of lipids throughout the body. And so, how is this actually done? Well, the liver, and again, this specifically is occurring in liver cells. That's why we just kind of say in the liver. The liver makes not only cholesterol, okay, but it also makes fatty acids. It also takes the fatty acids that it received from the chylomicron remnants, okay? And if there is, for example, too much triacylglyceride stored inside of the liver itself, in order to prevent toxicity from too much fat in the liver, it kind of breaks down those fats as well. And it takes all of this stuff and essentially packages it into this thing called a VLDL, very low density lipoprotein. Okay. And on top of this VLDL, it puts what we call an ApoB100 and an ApoE. All right. Okay so far? All right. Um, and there might be some ApoC2 on here as well. Okay. Um, yeah. Now, um, we have our VLDL. What do we do with this VLDL now? Well, guess what? I can take this VLDL molecule, right? And in fact, the liver does this, right? It takes the VLDL, it pushes it out into the circulation where it will go throughout, right, the circulation throughout the rest of the body, and it will distribute the fat towards what we call non-hepatic tissues, okay? Tissues other than the liver, all right? And those tissues, guess what? They have lipoprotein lipase on them, and they will be receiving free fatty acids and all kind of all the other stuff that comes with the breakdown of a triacylglycerol. But let's take a look at the VLDL pathway. So the VLDL, right? Like I said, it has the B100, the ApoE, and maybe some ApoC2 on it when it gets pushed out into the bloodstream, and it comes into contact with an HDL molecule in circulation that gives it even more ApoE and ApoC2. Okay. And when it receives these, right, it is now kind of eligible to go and interact with your tissues, right? And you can see right here the ApoC2 is, like we said, that apoprotein that activates the lipoprotein lipase, right? And it allows for the activation and interaction with the enzyme there. And that's going to cause, a hydrolysis of all of the kind of fatty acids in this VLDL. And after a couple rounds of this, the VLDL will become much smaller because, again, it's the things that are inside of it are getting less and less, and so it's getting smaller and smaller. It turns into this kind of intermediate molecule, if you will, okay, that sometimes may form and sometimes it may not form, okay? It just depends on the intensity of the cleavage that's occurring here. But this intermediate molecule is termed IDL, intermediate density lipoprotein, and it's this guy in green. Okay, notice how it is a bit smaller than our actual VLDL. Okay, um, and notice what has happened to it. It is actually lacking an ApoC2. Well, in the process of breaking down, we return ApoC2 into this HDL molecule. Okay, and this IDL has two kind of paths that it could take. Okay, so the IDL can either give back its ApoE, all right, to HDL and carry on as an IDL with just a B100 on its surface to the liver and be taken up by the liver by way of this thing called an LDL receptor. Okay? The reason why it will use the LDL receptor is because it has this ApoB100 protein on it. Okay? Or the IDL instead can become cleaved even further by way of lipoprotein lipase at the tissues, at non-hepatic tissues, and it can turn into LDL, okay? Low density lipoprotein, okay? Now, here's the thing. This VLDL was rather high in triacylglycerol content. IDL was a little bit lower, and the LDL we say is almost devoid of triacylglycerides. Okay, so very tiny amounts of you know triacylglyceride inside of this guy. But what does the LDL molecule do? Well, LDL, often termed your bad cholesterol, is actually quite important. All right, because without LDL, there's really no way of receiving cholesterol from the outside other than, of course, synthesizing it in the cell yourself. Okay, so LDL is the major way we get cholesterol from the bloodstream. And this is how it works. This LDL molecule will have LDL receptors on both non-hepatic tissues and hepatic tissues. And at non-hepatic tissues, okay, at non-hepatic tissues, it will deposit cholesterol, all right, by, again, endocytosis of this LDL molecule through these LDL receptors and the breaking down of all of that. And now we have cholesterol in these tissues, okay? And cholesterol, again, as you know, is important for many functions. It's important for hormone synthesis, right? It's important for incorporation into cell membrane, bile acids, a whole bunch of things, a whole bunch of things. Okay. And so some of these non-hepatic tissues might be tissues that require lots of cholesterol for synthesis of hormones. Okay. We don't know. It might be something like your adrenal gland. Okay. But that's what this LDL does. It can also go to the liver and it can be uptaken by the liver. And in fact, I have this note here for you. The liver is the major site of LDL removal. Okay, so 70 to 80% of circulating LDL is removed by the liver. Okay. And of course, what does that do? That gets you, once you kind of bring this guy here, right? Same process of receptor-mediated endocytosis, and you get these kind of cholesterols into the liver, okay? And this cholesterol in the liver may be used in multiple pathways, right? Again, it could be incorporated into the membrane. It could be put into the bile, um, um, or, it could be packaged into another VLDL for later, okay? But again, the important thing to understand is that we don't actually have in humans a method of oxidizing or breaking down cholesterol. Okay, we don't have it. This is the simplest form that we can have, this kind of multi-ringed system of cholesterol. This is the simplest form we can have. In humans, we do not have a way of breaking this down any further. And that is why the only ways of getting rid of cholesterol is by either putting it through bile or excreting it out um, in your feces, or making use of it somehow. All right. So that's the whole VLDL, IDL, and LDL. Um, I believe the stream cut out there for a second, but I think you guys caught the main explanation, right? Okay, good. So, any questions so far of this? Anything I can clarify for you? Okay, good. Again, if you have any questions at any point, feel free to drop it in the chat. So again, just as a brief review, right? This chylomicron was part of our exogenous lipid transport. The VLDL and the LIDL and IDL, they were all part of our endogenous lipid transport. And, you know, it's important to understand that the LDL and IDL are derived from VLDL. They're not like a separate moiety on their own. They are coming from the breakdown of VLDLs and the breakdown of IDLs, okay? Um, okay. So how does trans fat raise LDL and lower HDL? Um, is that mentioned? Because there's not a direct relationship between trans fat raising your LDL and lowering HDL. Um, there is a kind of correlation that exists that says um, unsaturated fats in general, so things like trans fat is an unsaturated fat, right? As long as you have a kind of double bond inside of your fatty acid, that is unsaturated, but it could either be trans or cis, right? There is an interesting thing that is present where LDL molecules that inside of them have a lot of unsaturated fats are more easily oxidized. And if they're more easily oxidized, then they can be more pathogenic, right? So those are the ones, those, you know, uh, oxidized LDLs are the bad ones. Those are the ones that are contributing towards things like atherosclerosis and whatnot. And the LDL molecules that are not oxidized, they're, well, benign. Okay. And so having these unsaturated fats can increase your um, probability of atherosclerosis by way of making your LDLs more susceptible to being oxidized and then more susceptible to kind of leading to atherosclerosis. And down the line through some regulatory mechanisms, you might change the ratio of LDL and HDL because of this. Okay, does that kind of answer your question? Hopefully. Okay. Again, in general, unsaturated fats, because of just the way their chemistry works, they're more reactive than something like a saturated fat, right? So they're more easily um, reacted with. Okay. The role of PCSK9. Absolutely, I can go over the role of PCSK9. So, but before we do that, very quickly, do you guys want me to um, finish the HDL, or are we okay with HDL? Because the HDL is Okay, yeah. So we'll finish HDL and then we'll go on to the PCSK9. So HDL, right, is our um, high density lipoprotein. And of course, the names, their densities, they're really just related to the lipid to protein ratio. As you can see, there is more protein on this guy than there is lipid. And so, well, relatively more as compared to at least this guy or this guy. And for that reason, the density of this molecule is a bit higher as the others. And HDL participates in a pathway called reverse cholesterol transport, where essentially, let me kind of bring this up here. Essentially, HDL, you know, it's made in the liver, but it can also be made a little bit in the small intestines. Let me kind of bring this up here. Um, so HDL, it's made mostly in the liver, but a little bit also in the small intestine. And once it is kind of produced, it will go towards your tissues, right, and pick up cholesterol, excess cholesterol, if you will. Okay. And each LDL will go through approximately 7 cycles, all right, before coming back to the liver and getting degraded. 7 cycles of what? 7 cycles of reverse cholesterol transport. Okay. So watch. LDL, HDL gets made, HDL goes to the tissue, picks up cholesterol. HDL continues, right? Goes to the liver, dumps off the cholesterol, continues back to the tissue, and so on, repeating the cycle 7 times before getting degraded, going back to the liver and getting degraded. Okay? And HDL has a lifespan of approximately 2 days. All right? Again, these are all kind of approximate numbers, right? Um, yeah. So, I do want to um, check something very quickly. Okay, that's fine. So, okay. So you know what? Let's do it this way. Once the HDL is made, it is in what we call a nascent form or a discoidal form. Okay, it is kind of like flat in shape, right? And the reason is because it's not really filled with anything, right? It is kind of just phospholipids at the moment, right? A little bit of kind of cholesterol in it, but not much of anything, really, right? So we have this nascent discoidal HDL at first, and it has apoproteins of C2, ApoE, and ApoA1 in it. And if you recall, we saw this HDL kind of interacting with other lipoproteins in the bloodstream, right? And it does that throughout its kind of life cycle, and donates some of these ApoEs and ApoC2s and ApoA1s on it, right? We saw this multiple times. But this guy right here will make its way to non-hepatic tissues, right? So you're given examples here of adipose, of muscle, arterial walls, right? That's an important one. Arterial walls, why is that an important one? Because you want to prevent atherosclerosis, right? And so more HDLs performing their HDL cycle is beneficial for you to prevent an atherosclerotic plaque buildup, right? Preventing that high kind of blood pressure that may arise from that. But once it makes its way to the tissues, these non-hepatic tissues, it interacts with this specific receptor on the tissue is called an ABCA1. What does the A1 stand for? Well, the A1 stands for the fact that it interacts with the ApoA1 protein. Okay? And the ABC, it just refers to the kind of transporter that it is. Um, it stands for ATP binding cassette protein transporter. It's just a kind of transporter. You don't really have to worry about it too much. Just know that the name is ABCA1. And it interacts with this ApoA1 protein, and it allows for the diffusion or movement, I should say, of this cholesterol into this HDL molecule. Now, the HDL molecule inside of it has a very important enzyme called LCAT. Now, what this LCAT does is it's really interesting. This LCAT converts the cholesterol that's coming in into a cholesterol ester. The reason it does this is because remember how things like to go from a high to low diffusion gradient, right? Well, this LCAT enzyme, by turning this cholesterol into a cholesterol ester, maintains a continuous diffusion gradient for cholesterol out of the tissue. How is it doing this? Well, imagine this. If this enzyme wasn't there, eventually the amount of cholesterol that is coming in from the tissue into this HDL will start to kind of reverse, right? And the gradient would start to reverse. LCAT prevents this by taking that cholesterol and turning it into a cholesterol ester and maintaining a low concentration of cholesterol in the HDL to allow for continual diffusion out of the tissue. Okay? Does that make sense? Because that is important. Okay. So once it does that, um, and the enzyme LCAT is called, I'll write it for you, lecithin. Um, wait, I do want to make sure to get this right. Lecithin cholesterol acyl transferase. And if you know your phospholipids, you know that lecithin is a phospholipid. And so I don't want to get too detailed here, but essentially what this enzyme is doing is it's taking a fatty acid from one of the phospholipids and it's transferring that fatty acid, otherwise called an acyl um, group. It's transferring that to your cholesterol to make a cholesterol ester, because all a cholesterol ester is your regular cholesterol plus a fatty acid attached to it. Okay, so that's what this enzyme is doing. It's pretty interesting. Um, but eventually, enough cholesterol will come into the tissue, come into the HDL from the tissue, that it makes this cholesterol, this HDL molecule spherical, and then we say that it has now matured. Okay, it is a mature spherical HDL, very high in cholesterol and cholesterol ester content. Okay, so look at it. It has this, this, and this, very high. Um, and it is over here where the HDL will um, go into the actual liver, okay? And by interacting with this specific receptor on the liver called SRB1, scavenger receptor B1, by interacting with that guy, and I've omitted the labels, but it still has ApoA1, it still has ApoE, and it still has ApoC2. It will interact with the liver's scavenger receptor B1 and deposit all of the cholesterols and go off to do another cycle, right? It will do this until it becomes again kind of nascent and discoidal in shape and go off and do another cycle, right? In fact, we know it will go and do 7 cycles, okay? So that's the HCl cycle. And one last thing that I do want to mention about this is that remember, again, this HCl in the um, circulation will interact with things like your VLDLs, like your chylomicrons, right? And so not only is it exchanging aproproteins between the VLDL and the chylomicrons, just like we saw here, right, or here, It is also exchanging content. All right, so there is enzymes called cholesterol ester transfer proteins and phospholipid transfer proteins, and the the um the HDL will always give away its cholesterol. Why does it want to give it away? Because um, it wants to maintain enough space inside of itself to go and pick up more cholesterol from the tissue. So, by way of this cholesterol ester transfer protein, it will transfer the cholesterol esters to either a VLDL or a chylomicron, and it will pick up phospholipids instead from these guys. Okay? And of course, again, in this interactions, apoproteins are being transferred as well. Okay. Um, any questions about any of the kind of transport stuff for lipid transport? Any clarifications you may need, anything at all? Okay. Are we okay to move on to PCSK9 and its role? Okay. Let's do that. So here's PCSK9. Remember how we had the LDL receptor, right? We had the LDL receptor at the level of the tissues, right? Extrahepatic tissues, and we had the LDL receptor at the actual hepatic tissues, okay? So PCSK9, all right, is a protein, okay? It's a protein made by the liver mainly, okay? Other tissues can make it, but it's, mainly made by the liver. And in order to understand what it does, we have to understand what the LDL receptor does. So I have this kind of depiction here. LDL receptor, right? So that is the same as this guy and this guy that we were showing before. Same LDL receptor, just kind of a different depiction. We know that the LDL receptor will bind the molecule of LDL. It will get internalized, right? That's the process of endocytosis. And the LDL particle will separate from the LDL receptor, and the LDL particle will be degraded and release, cholesterol content and all that stuff, while the LDL receptor will be recycled back to the membrane, ready to kind of pick up another LDL particle. What does PCSK9 do? Again, PCSK9 is a protein that is made by the liver and it's secreted, okay, meaning it is kind of, pushed out of the liver into um, the bloodstream. Um, and in different tissues, PCSK9 can go and bind to LDL receptors, all right? And once it binds to this LDL receptor, look at it. Here's PCSK9 in green. PCSK9. Once it binds to the receptor, right? The next time an LDL molecule comes and binds to our LDL receptor, both the receptor, the PCSK9, and the LDL will get endocytosed in, just like last time, except now there's PCSK9. And this time, what actually ends up occurring is instead of separating the LDL and the LDL receptor from one another and recycling the LDL receptor, what I do is this PCSK9, it has caused the degradation of both the LDL and the LDL receptor. Okay? So it actually prevents LDL receptor recycling. Why is that a good thing for us? Because if we are to, go with the idea that LDL uh, is a bad cholesterol, right, and too much LDL is bad for us, which is true, then how does LDL kind of exhibit its bad effects? Well, it does this by binding to its receptor and then coming into the cell and releasing the cholesterol in the cell, and then its receptor going back on the membrane, ready to bind another thing. But if I lower the number of receptors on the cell surface membrane, then by logic, I have less ability to bind this LDL, and this LDL will just kind of circulate harmlessly because the only way it can exert its effect is by binding to its receptor. Okay? And so PCSK9 is going to do that. All right? It's going to kind of lower this um, presence of our LDL receptors by kind of allowing it to be degraded instead of being recycled. Um, okay. Any questions? Yeah, so when PCSK9 binds to the receptor, the LDL receptor, the next time the LDL receptor binds an LDL molecule, it will cause a degradation of both of them. And that way the receptor can, it won't get recycled back because in reality, this receptor has a lifespan of 20 hours, meaning it stays on the membrane for 20 hours and it gets recycled every 10 minutes. All right. So if it gets recycled every 10 minutes, it's ready to pick up another LDL. But PCSK9, as a protein that we synthesize on our own, prevents this by kind of degrading this um, LDL receptor. And, it's actually there as a kind of a negative feedback mechanism. For example, let me ask you guys a question. If we know that the cell can make cholesterol on its own, right, through our little pathway that we've talked about in the class, right? But do you think that if the cell is making enough cholesterol on its own, do you think it wants to take up cholesterol from the bloodstream by way of LDL? What do you think? If the cell already makes enough cholesterol, would it want to pick up more cholesterol from the bloodstream? What do we think? The answer, of course, would be no. Exactly. Right? Why would it be no? Because it is just like everything else in the human body, a negative feedback mechanism. Okay? We always want to stay at a kind of homeostasis, right? We don't want to be too far away from it. And so if I have enough cholesterol already, why would I take more cholesterol in? That doesn't make sense. So as a negative feedback mechanism, this PCSK9 is there to kind of prevent us from doing that. Now, pharmacologically, we can use drugs called PCSK9 inhibitors to lower circulating LDL, levels. Okay, and what I mean by this is LDL levels, right? People go get their blood tested and they see a high LDL level and they start, going crazy. They're like, "Oh my God, what do I do?" Um, what you know, somebody might get prescribed, right, on top of multiple other drugs like statins and stuff, is something called a PCSK9 inhibitor. And the logic works as such. If I prevent PCSK9 from binding to the LDL receptor, right, then it won't get degraded and it will continue to recycle and more of it will stay on the membrane, right? So a PCSK9 inhibitor works by blocking PCSK9 from binding to LDL receptor, preventing this degradation process and leaving the LDL receptor on the surface more. And guess what? If it does that, then more of the LDL particle will be taken up from the bloodstream and your circulating levels of LDL are lower. Okay? So that's that. Okay? That is the kind of idea behind a PCSK9 and a PCSK9 inhibitor. All right? Any questions of this? Any clarifications I can make for you? No problem at all. My pleasure. Okay. He might ask about, so I'm sure you're familiar with the way he asks questions by now, right? He likes to ask very integrative questions, right? Questions that test your um, knowledge of things overall. Um, and so with these pathways, there really isn't kind of, you know, too much that he can change because the pathway is the pathway. But you do have to know the details of this and this, okay? Um, but questions such as the one that he did in class with you guys, those are extremely similar. I mean, extremely similar to what he will ask on the test, okay? That same level of thinking required to answer those questions and that same level of detail is what you're going to see. With his exams, it really is more so paying attention to detail more than like what is the specific kind of type of questions because the content, once you get the content, that's it. It really is paying attention to his wording and his what is the question that he says is specifically asking me because he is a very big stickler on details. So that is one of the best advice I can give you guys for this because even on the first midterm, a lot of kind of errors I was seeing um, was with people either not reading the question carefully enough, and despite them knowing the answer, because the question is worded sometimes a bit more tricky, um, people either doubt themselves or something, and they put the wrong answer. So this is my best advice to you to read it very carefully. And I know it is a little bit hard with the amount of time given, but I think that is probably one of the best advice I can give you. Um, yeah. So let me see anything more specific I can help you with. Definitely be familiar with the mechanism of PCSK9 inhibiting this LDL receptor and how might giving a PCSK9 inhibitor be used as a therapeutic option. Be comfortable with that idea um, of how that actually works, right? Just like what we talked about. Um, yeah. Um, what else can I tell you? Um, in terms of proteins, so there's many. So there's many for proteins. There's many kind of mnemonics out there that are available um, for remembering this kind of stuff. Answer me this question: does she give you guys the structure or no? I want to be. My advice is going to change depending on whether or not if she gives you guys the structure. Does she give you guys the structure of the amino acids? like on the test. So, but like without the names or no? So you don't have to remember the structure, but you have to remember the names and what type they are. Right. But you're not given like this on the exam without like the names, right? Like almost like kind of like a cheat sheet of the structure. You're not given that. Okay. That's yeah, that's okay. Okay. Okay, good. So that's what I wanted to know. So yeah, that again, that's annoying because that makes it a little bit harder because the structure really does help you. Um, I used to come up with kind of mnemonics. Okay, there are many that you can find online for yourself. Um, I kind of used to group them into their thing, like non-polar amino acids. I used to group them with their one-letter codes. If you want, I can show you the one-letter codes. A lot of them are easy to remember. And I'll give you advice if this is how I did it. A lot of them are just their first name. So glycine obviously is a G, alanine's an A, valine's a V, leucine's an L, isoleucine's an I, methionine M, phenylalanine. Phenylalanine, I like to remember because it makes a F sound, is F. Tryptophan, it's, I just kind of remember it's a W because there's nothing else that's a W. Proline obviously is a P. Aspartic acid, again, that's one of those weird ones you have to just kind of remember it's a D. Glutamate ends in an E. I like to remember it's E. Arginine makes an R sound, so I say it's R. Lysine, I like to see it like this. This L, if I put a nice little line through it here, looks like a K. So its one letter code would be K. Histidine, H. Um, what else? Serine, S, threonine, T, cysteine, C. So again, you see a lot of them are exactly what their name is. Like their one letter code is exactly their name. Tyrosine, only one that has like a Y here. So it's Y. Asparagine has this N. I like to remember for this. Um, um, Glutamine. I like to again play around with the letters. So glutamine kind of looks like a Q if I do this, so it's a Q. Okay, but this is how I used to kind of remember their their specific properties, right? I would just come up with mnemonics that I would remember based off of their specific one letter code, right? I would learn the one letter code, and then once I knew the one letter code, I could just apply it here. And I'm telling you right now, the best way to do this is by grabbing a blank piece of paper or opening up a new slide for yourself on your little tablet and just start writing. Just start writing. Okay, just start writing and you know memorizing it because generally I would give the advice of kind of remember the structure because the structure does really help you. But in this case, since you're not given the structure, you just have to write it down. You just have to write it down. So I would remember like gap v lim for non-polar, allopathic, and I would remember the one letter codes, right? Uh, let's see. So G, glycine, A, alanine, P, proline, V, valine, L, leucine, I, isoleucine, and M, methionine. Every single one of these, their one letter code is the same as their name. Every single one of them. Okay. And you might be saying, well, how do you not confuse something like glycine and glutamine as being G? Because I know that glutamine has that kind of um, this. So I like to do this for glutamine and say that it's Q. And you might say, okay, how do you know it's not glutamate? Because I remember that glutamate ends with an E and it must be E. Okay, so again, it really depends on you. and what you like to do with these. These were like always personalized, even in biochemistry, super personalized. Um, People have different ways. This is my way of doing it, so this is my recommendation. But a general recommendation I can make to you is always start by writing all of these down multiple times so you can just start becoming familiar with the names. All right. Don't just look at them and start saying them out loud. That's going to do you no good. Write them down for yourself. Okay. The positive charge ones and the, negative charge ones, I find they're the easiest to remember. Positive charge, always going to be arginine, histidine, and lysine. Okay. So again, lysine, histidine, and arginine. Again, arginine, that R makes that R sound there, right? The negative charges, D and E, it's going to be aspartic acid and glutamate. Oh, and by the way, one thing I do want to mention, because a lot of people do mess this up, is you can write these amino acids, well, I should say that you can write these two amino acids as either aspartate. Okay, so this can be either written as aspartate or aspartic acid. These two are the same thing. Same thing. This can be glutamate or glutamic acid. Same thing. Okay. Um, I remember, last year there was a couple of people who didn't. Uh, I guess it wasn't specified in lecture or something, and so people got tripped up by that. So um, those are the same thing. They're referring to the same name. Um, but again, that is my kind of thing. Definitely know your branch chain amino acids. There's going to be a question on that. And that's not too bad, right? So it's kind of like ILV, okay? Isoleucine, leucine, valine, right? Those are your branch chain amino acids. Um, you know, the polar ones, again, come up with mnemonics for yourself. Come up with this again, this is the best way I did it. You can write stuff like this down. You can, I don't know, arrange these letters to make your own mnemonic if you want, something that you will remember. And yeah, right. I had friends of mine who did that. Um, so yeah, okay, that is my best advice. The importance of branched chain amino acids. In what sense? So branched chain amino acids, especially leucine, is extremely important for stimulating muscle protein synthesis. And you need about 3 grams of leucine in order to achieve optimal levels of muscle protein synthesis. And depending on the kind of protein you're having, there will be a different amount of that protein that you have to, uh, ingest. So, for example, if you're having something like whey protein, you might only need like 23 grams of whey protein to get 3 grams of leucine. Um, but in something like, I don't know, wheat, you might need like 46 grams to get that same 3 grams of leucine, right? And so it's uh, the importance of these branched chain amino acids, specifically leucine, is that they are very important in activating muscle protein synthesis. Okay, anabolic pathways in the muscle that can lead to the synthesis of protein. They're very important for that. At a molecular level, they activate this thing called mTORC1, if you're curious. Um, and so having adequate amount of protein that contains enough branched chain amino acids is important, and the type of protein you ingest matters as well, right? So, like I mentioned, something like wheat, right? You're going to have to get 46 grams of wheat to get 3 grams of leucine, which again is that threshold required for optimal muscle protein synthesis. And for something like a whey protein, you only need what, 23 grams, 26 grams, right? So there's a 20-gram difference between the ingestion of that protein. Um, so yeah, let me know if you want me to, explain it in a different way or something that I didn't cover. When explaining the water classification of amino acids, she mentioned that tyrosine aromatic, but also polar. Mary, absolutely, I can do that. So here is our molecule of tyrosine. So um, have you ever heard of a benzene before? Here's a benzene. Do you see that benzene in this molecule of tyrosine, in this amino acid of tyrosine? I do. I do. So it's right here. I want to make something clear. Again, this is probably not explained because it doesn't really need to be explained. Aromatic is a concept in organic chemistry which you do not need to know, but people make the mistake of assuming that anything that has a ring must be aromatic, and that's not necessarily true. But I will give you one thing that you can remember. Anything that has a benzene in it is aromatic. Let's prove that to ourselves. Phenylalanine has a benzene in it. It is aromatic. Tryptophan has a benzene in it. It is aromatic. Tyrosine has a benzene in it. It is aromatic. It has to do with organic chemistry concepts, but for you, it is sufficient to understand that anything that has a benzene in it will be aromatic. So this is why tyrosine is aromatic. But why is it polar? Well, polar, again, is another chemistry concept. It just refers to separation of charge. And oxygen and this H here, this group is very polar, okay? And it means that it can interact with water quite nicely. So it is water soluble, all right? And because of this OH group that it is polar, okay? So tyrosine is both aromatic because it has this benzene, and it is polar because it has this OH group, okay? Make sense? Okay. Any other questions that I can maybe kind of assist with your understanding or whatnot? Mm, um, uh, from the textbook slash lectures, I'm under the impression that unsaturated fats are more associated with. So, this is a thing, John, you're bringing up an interesting point. So, I'll tell you this, okay? Without, getting into a conspiracy. Um, oh, Saturated fats have historically been thought and associated with cardiovascular disease. Okay. Um, but more recent evidence suggests that unsaturated fats, due to their reactivity, um, increase cardiovascular disease risk. Okay. And multiple of kind of, you know, the I'm trying to find the page here for you. Multiple of the evidence that he brings up suggests similar things. Um, So I will kind of refer you to this. I don't know if you can see it. I think you can see it. There you go. That page right there. So that right there is showing you the association, and this is completely association, it's just correlation, between the consumption of vegetable oil, which is mainly unsaturated fats, and saturated fats consumption, and heart disease deaths. And what he's trying to suggest here is that despite what um, the media and big oil might have you believe over the years, is that saturated fat consumption has remained relatively constant throughout the years, and yet heart disease deaths has seemed to gone up. And so it doesn't really seem to track, right? Obviously, correlation is not causation, but if we were to make the claim that saturated fat is causing heart disease and heart disease deaths have gone up over the years, but saturated fat consumption has stayed the same, it doesn't really seem to track. What does seem to track better with that is unsaturated fat consumption, in that unsaturated fat consumption has gone up over the years, and that trend seems to be more similar to the heart disease death trend as well. Okay, so this is his philosophy. Really, is that he gives you the information and you can interpret it whatever way you really want. But in reality, on the test, you won't ever really be asked to kind of, you know, what is your take on unsaturated versus saturated, right? It's whatever he's taught you that he will test you on. Okay. Um, for example, he might ask you, why is it that an unsaturated fat is less unhealthy to cook with than a saturated fat, right? And give a couple of options. And the reason is because unsaturated fats, um, when you heat them up, right, cause the release of these toxic chemicals that saturated fats do not. And again, that has to just do with their chemical composition. Okay. So it is a bit of a controversial topic, um, but such is the nature of many things in nutrition. I hope that makes sense. Yes, I am invigilating tomorrow. Tomorrow, right? Yeah, it is Sunday. Um, yeah, yeah. Okay. Any other question with protein? I feel like we went through a lot of lipids, and that's completely understandable because lipids was the hardest part, I think. Um, but any questions about the protein section? Other than obviously, how to go through the amino acids and stuff. And I would be quite comfortable with that, guys, because she does like to ask those kind of questions of which of the following is a polar amino acid, and gives you like five options, and only one of them, of course, is correct, right? So I would be very comfortable with that. I would focus on writing those down for yourself, and if you have a mnemonic when you get your test paper, you can just write down your little mnemonic so you remember it, right? Um, yeah, I do recommend this kind of stuff. Um, yeah, yeah. What time is it here? It is 12:40, so it's been like an hour 40, I think. Right? Yeah, it's been an hour 40. If nobody else has any questions, we can call it a day here, and I will let you guys get back to happy studying. Okay? What do we think? Are we good? Good. No problem. No problem, guys. No problem. Thank you guys for showing up. I mean, I didn't know if anybody was gonna show up. I just kind of had the idea, um, and thought maybe it would be helpful. Um, yeah, amazing, guys. Amazing. Thank you so much for coming. Okay, and best of luck tomorrow. I'll see you tomorrow. Um, you know, yeah. Bye bye, guys. Thank you.